Alright, sorry about that. So we're gonna go back to prokaryotic cells. So uh, the prokaryotic cells are very simple compared to eukaryotic cells. So prokaryotic cells can be considered like bacteria, right? Bacteria. So bacteria. Alright, sorry about that. So what makes prokaryotics different than eukaryotics? So prokaryotics, they have no DNA. Instead, their DNA is No, not bad. They have no nucleus. So their DNA is circular, and they concentrate in an area known as the nuclear region, right? And they got no membrane-bound organelles as well. And a lot of bacteria have flagella mycelium that helps them basically swim around. So the prokaryotic domains are archaea, bacteria, and eukarya. So yeah. The archaea is uh, very similar to bacteria, but they contain genes, which bacteria don't. But they also have similar metabolic pathways to eukaryotics, right? And what else am I gonna go higher? Oh, sorry about that, guys. So we're just gonna go and erase all this. That's cool. And archaea are often considered to be pretty extremophiles, so they can live in pretty harsh stuff like those little volcano vents down deep in the ocean. And they use chemosynthesis, which is instead of photosynthesis like a lot of plants do, they use chemical sources of energy. And eukaryotics, they're similar to eukaryotics by they can start translation with methionine, and they have similar processes with RNA polymerase and histones. Well, similar to bacteria, they have similar circular chromosomes, and they divide by binary fission or like through asexual reproduction. So now we're mainly going to talk about bacteria now. Bacteria. All right. So wow, that's just how complicated this stuff is. So we might need to split this up, but yeah. Uh, Alright, so bacteria contains the they have cell membrane, of course. They got cytoplasm, and they even have some of them even have flagella. So bacteria is by far the most abundant out of all the cells on the planet. They outnumber both plant and animal cells by like something like 10 to 1 or something. So or I don't know if that's the right number, but it's a lot. It's way more than animal and plant cells. It's everywhere. We have bacteria living in us. A lot of the food we eat has bacteria, like milk and cheese. So there's a lot of classification of bacteria. It could be like mutualistic symbiotes, right? Where both the human and bacteria benefit. Like for example, we have bacteria in our gut and living all over our body. And there's also pathogenesis, is where the bacteria can cause an infection or disease. Okay, now there's three different types of bacteria shape. There's this cocky shape. So it's like circle shape like this, cock shape. Then we got bacilli, which is like rod shape, right? Rod shape. And then we got spirally, which is more uh like spiral shaped. Yeah, just like the main three types. Anyway, so now we're gonna talk about aerobes and anaerobes. So there's four main types. So we're gonna go over what an anaerobe is first. So anaerobes are basically things that don't need sorry, that don't need oxygen for cellular metabolism, right? Well, obligate anaerobes, they cannot survive in oxygen at all. So if you give them oxygen, it kind of results to the production of a lot of reactive things going on inside them that eventually leads to them dying. So yeah, not gonna go too deep into that, but that's how it goes. And then we have obligate aerobes, so they need oxygen for metabolism. And some bacteria can actually switch between aerobic and anaerobic metabolism, and that's called a facultative anaerobe. And there's also aerotolerant anaerobes, so they can't use oxygen for it. They don't use oxygen for metabolism, but they're not harmed by it, like how uh, obligate anaerobes are. So yeah, that's kind of goes for that. And so prokaryotic cell structure, it's a bit complicated. All right, it's a bit complicated. So it goes like this, right? And then usually it goes circular DNA. Like DNA protects all DNA, stuff like that. And they usually got some kind of like flagella sticking out of it, right? You get the cell wall, and of course, like usual, it has a cell membrane. Okay, they're considered to be single cell organisms, and all those little things here are ribosomes. Well, this is the nuclear region where the DNA exists. This is the flagellum. I made this a little too big. It should be a lot smaller, but whatever. So there's a difference between gram and gram-negative bacteria. So, like usual, the phospholipid is still composed of, the hydrophobic and hydrophilic portions. So it's actually a pretty interesting way of classifying the cell wall to gram-positive and gram-negative. So gram-positive, their wall is more thick and it's composed of peptidoglycan and liptocholic acid, while the gram-negative contains phospholipids instead and lipopolysaccharides. So a way for scientists to basically study this is basically grab a crystal violet stain dye, and then they stain it, and then they eventually stain it again with a substance called safranin. So if you ever took bio class and did a lab, you probably did this before at some point. It's really cool, but also interesting. So if it was gram positive, it'll turn out to be just crystal vanilla stain, only crystal violet stain, because it's really sick, and the saffron won't be able to bleed through. Or gram negative, it'll turn pink red because because it stains the thin walls of it, right? So that'll be gram negative. Alright, anyways, going on next. I'm not gonna go too deep on the flagella structure and stuff. There's a lot of different body parts like the filament, basal body, and the hook, all designed to help the body move. What else? I'm not gonna go too deep into other stuff. There's binary fission, of course. Binary fission, genetic recombination, transformation, and conjugation. So the bacteria has a lot of ways where they basically reproduce. So, again, not gonna go too deep into it. All right. So, transformation is where it's the acquisition of exogenous material that can be integrated into bacteria genome. So, uh, how do I say this? How do I do this? It's basically like the basic form of binary fission. That's all I can say. So, conjugation is the transfer of genetic material from one bacteriophage to another via a conjugation bridge. So, it's basically cell sex. Yeah, bacteria sex. And they use this thing known as the pilus in order to basically inject its DNA into another cell. And yeah. And then transduction is through viruses, through a bacteriophage. So how it works is that the bacteria, the virus, also known as the vector, will inject its DNA into a bacteria. Then that DNA would basically replicate itself into the bacteria, right? And it will keep replicating until it forms something called lyse, and it will burst out of the cell. And that virus will come out and start injecting its DNA from a bacterium, and then that DNA still has parts of bacterium genome in it, and it will eventually be part of the infected a new bacterium's genome. So, and eventually it'll build up again into like something called like a lysogenic cycle. It can stay dormant for the years and years, and eventually it can be activated, where it eventually go back to a lytic cycle, and go back to the process of how the cell bursts, keep multiplying in there, and then it bursts through the cell through a lyse, and then it goes on the process, and then this virus keeps spreading all over the cells, just like that. So, bacteria, the most common form of way of these ways bacteria form is through conjugation. That's the most common way of bacteria spreading or reproducing. Now, there's 4 phases to bacteria growth, so you can kind of look at it through a chart like this, right? So we got boop, 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 right? So gonna split up like this. This phase right here will be known as like the lag phase, where the bacteria is just trying to adapt to its environment still. Then this phase here is where the cell just jumps up and starts growing like crazy. But here is where their resources are kind of just at the same level, and they're kind of just stagnant. It's also known as the what's it called again? The stationary phase. Then when the cell starts to die down when it depletes its resources, also known as the death phase. Alright, so um now we got what else we got here? Now, viruses. I was just reading about this. It's a lot complicated, but I'm sure you guys all seen this picture of a virus. Right, so get rid of this. So because this give us a hexagon like this. That and virus like that. Right? So a virus, but inside the thing, it's like kind of like hexagonal here. And inside here, where be the DNA is, nucleic acid, and also where the capsid is, alright, protein. And then here would be called the tail sheaths, and it looks like a little coil if you look at it carefully. And then these are we known as the tail fibers, So this is what a typical virus look like a bacteriophage. So a bacteriophage is a type of virus that infects bacteria. So what's so special about the virus? So it's not considered a living thing because it violates one of the 4 tenets of cellular theory. It's considered acellular, which means it's not made of discrete cells and it can't split into other cells. It requires another host to do it for it, right? So they're called obligate intracellular parasites because they can't reproduce independently. They have no way to do that. So they need a host cells because they don't have any ribosomes inside them to carry out any sort of protein synthesis. So yeah, this is a bacteriophage, so it reproduces through transduction, right? All right, and now there's viral genomes. They also have their positive and negative sense. So a positive sense means it'll be translated by the host cell, and then the negative sense, which means it requires the complementary strand to be synthesized by RNA polymerase before translation. So I'm not gonna go too deep on transcription and translation. So there you have it. And then you got stuff like retroviruses, where they only have a single strand of RNA genome and the complementary DNA strand is made using reverse transcriptase. So, for example, it's a lot harder to cure, basically, because they eventually become embedded into the genome. Like example would be like human immunodeficiency virus or HIV. Now we have an infection, or how this works is that it basically attaches itself to certain receptors, and then They enter the cell through their membrane, and then they start injecting stuff into the genome of the cell. So yeah. Anyways, now we're gonna go over to how the cell actually, how a virus will actually um, replicate. So a virus, they reproduce by replicating and translating genetic material using the host cell's ribosomes, tRNA, amino acid, and enzymes. Basically, depleting the cell down to its resources. So there's positive sense RNA viruses. They stay in the cytoplasm where it's directly translated to proteins by the host cell's ribosomes. While negative sense RNA viruses, they require synthesis of a complementary RNA strand via RNA replicase, which then translates eventually to protein. So, yeah, not go too deep into that. Progeny release, there's quite a bit of them. So, there's one that's where they just keep replicating inside a cell until a lyse happens and they just burst out the cell. And then they got one where uh, what else? They got one where the virus can just leave the body just through the plasma membrane. That's called extrusion. And when that happens, it will usually call a productive cycle. So the virus goes through two main life cycles. One is called the lytic cycle and the lysogenic cycle. The lytic cycle is a bacteriophage that tries to maximizes the cell's uh, like resources to maximizes its survival. So it'll just keep using all the cell's resources until it eventually dies out. And then when it dies out, it bursts out the cell and all the viruses go everywhere. So it's considered virulent. And now we have lysogenic cycle, which is similar to lytic cycle, but it integrates with the host genome as a provirus or a prophase. So it reproduces along with the cell. So it becomes part of the cell's genome. So it can actually stay inside of a cell for like dormant for like years and years until eventually it gets activated by a stimulus, and it goes back to the lytic cycle where it keeps replicating inside a cell until eventually it bursts through a lyse and it all spills out everywhere. Right now, there's two types of things that are known as prions and viroids. So they're basically like small particles that can cause disease when they're under certain circumstances, right? So for example, prions, they cause infections through proteins, right? They misfold your proteins. They cause disease by triggering the misfolding proteins, and they cause reduction in solubility of your proteins, which interact with cell function, right? So, and then they basically convert your alpha helicase structure to a beta pleated sheet. So that's really important to know, this part. They convert your alpha helical structure to a beta pleated sheet. Well, viroids, they basically just turn off your cells like genomes or genes. They the pathogens are a small circle of complement RNAs can be turned off genes, resulting in metabolic and structural instability, which eventually leads to cell death. So, yeah, it's kind of what's your question so far. Eukaryotic and prokaryotic and cells, as well as cell theory, the organelles, how they all multiply. We're gonna go through the next time about how eukaryotic cells reproduce. So, prokaryotic cells, they reproduce pretty simple. Uh, Bacteria through transduction, binary fission, conjunction, and transformation. Right. But bacteria, viruses, they do it through true lytic and lysogenic cycle, and they're able to infect other things by injecting their DNA into other healthy cells, right? So either bacteriophage or just a regular virus. So yeah, that's kind of it. So good stuff, good stuff. Gonna keep grinding out the MCAT. Going through. We're pretty into it. My goal right now is to completely finish the biology book, then move on to chemistry, and then biology, and then move on to biochemistry. And after done with those two, we're gonna go into chemistry, and then we go to organic chemistry. That's gonna be a one whole set to know by itself, right? Then we're gonna go into behavioral science, right? It's behavior. Oh, actually, I'm not sure. We'll cross that bridge when we get there. So yeah, thank you guys for watching. If you got this far, and see you on the next trip.